According to the U.S. Surgeon General's report on youth and tobacco use, over 86.9% of adults who smoke cigarettes daily report that they had their first cigarette before the age of 18. The same report estimates that the average use of smoking initiation is just 15.3 years old. These numbers aren't just statistics, they show evidence of a critical developmental window. Adolescence is a period of intense neurobiological remodeling. Exposure to substances like nicotine during this vulnerable period can disrupt reward circuits and hippocampal learning systems, developmental processes in ways that have long-term behavioral consequences. In previous episodes, we examined one potential missing genetic link underlying nicotine vulnerability, or simply the alpha-6 subunit of the nicotinic acetylcholine receptor in the paper, Unraveling the Role of nAChRs, the neuronal alpha-6 nicotinic acetylcholine receptor subunit, by Yasmin Haji Haydari, Emily Castro, Shardad Lafipur, and Francis Leslie. We discussed how the alpha-6 containing receptors are strategically localized on dopamine neurons within the ventral tegmental area, positioning them to regulate nicotine reinforcement and addiction susceptibility. Today, we're going to shift our focus to another subunit within the same receptor complex, the alpha-2 subunit, which is encoded by the nAChRs gene. Welcome to the Translational Addiction Podcast, where we explore innovative research from the Lafipur Lab at UC Irvine, dedicated to advancing translational addiction science and developing more effective treatment strategies. I'm your host, Ishva Patel. I'm a second-year business and biology major at UC Irvine, and I'd also like to welcome our host.
Hi, my name is Arshia Sen, and I'm a first-year undergraduate student at UC Irvine. I'm studying biomedical engineering.
And hi, my name's Michaela Samorton. I'm a fourth-year undergrad at UC Irvine, and I'm studying neurobiology. So today, we'll be discussing the paper Deletion or Enhancement of Alpha-2 Nicotinic Acetylcholine Receptors, nChRNA2, which induces unique lifespan behavioral deficits, by Alicia Wells, Selena Mojica, Christy Fowler, and Shardad Lafipur. This paper is a primary research article, meaning it outlines original experimental data collected and analyzed by the authors. We will see the research question that was analyzed, the methods used, and the analysis techniques, and uncover new findings. The format is slightly different from what we've seen in our previous podcasts, where we have focused primarily on review articles. These do not present new experiments, but instead, they review papers and evaluate findings from multiple primary studies. To recap, the nicotinic acetylcholine receptors, or nAChRs, are ligand-gated ion channels activated by the endogenous or internal neurotransmitter acetylcholine, and by the exogenous or external ligands such as nicotine. These receptors initiate the flow of sodium and calcium ions, triggering an action potential, and can be found throughout both the central and peripheral nervous systems, and are critical regulators of many cognitive functions and withdrawal symptoms. Within the nAChRs complex, the alpha-2 subunit is crucial in hippocampus-dependent learning and memory, and the medial habenula interpeduncular nucleus, an important circuit in nicotine withdrawal. We could see this in previous studies that examined the alpha-2 and alpha-5 containing nAChRs, which showed that administration of mecamylamine, a nicotine antagonist, into the habenula or interpeduncular nucleus of the nicotine-dependent mice was sufficient to trigger withdrawal symptoms. Additionally, this nicotine withdrawal has been shown to enhance neuronal activity in both the interpeduncular nucleus and the dorsal hippocampus. However, in mice that lacked the alpha-2 subunit, there was no increase in neuronal activity from withdrawal. To build off of this, in a prior study by the Lafipur Lab, the alpha-2 containing nicotinic acetylcholine receptors were manipulated in two ways. First, by deleting the nAChRNA2 gene, and second, by enhancing the nAChRNA2 gene. Researchers were actually able to examine how nicotine influences learning and memory. So the results of this study were very shocking, showing how age, sex, and bidirectional manipulation of the alpha-2 gene reveal differences in learning and memory. Furthermore, this prior study also revealed how these same differences could impact nicotine reinforcement. Overall, there was a clear evidence of age and sex differences in these two areas. So, given these age and sex differences, the present study sought to expand on this investigation by looking at how the genetic manipulation of the nAChR gene alters hippocampal-dependent learning across age and sex, along with nicotine reinforcement. As previously mentioned, today we will dissect this paper by outlining the methods used in the study, the analytical techniques, and the major results of the paper.
In this study, the researchers used 3 mouse models, which were each designed to isolate a different role of the alpha-2 receptor. First were the wild-type mice, which were treated as a control group. This means the mice had normal alpha-2 receptor function. These included both adolescent and adult male and female mice. The second group was the knockout mice, where the nAChRs gene was deleted. This means that these mice didn't produce the alpha-2 receptor at all, allowing researchers to observe what behaviors change when the receptor is missing, specifically behavior involving fear learning, extinction, and nicotine reinforcement. These included both adolescent and adult male and female mice. The third group was the hyperexcitable L9S mutant mice, in which a point mutation makes the nicotinic acetylcholine receptor much more sensitive to both endogenous and exogenous ligands, such as acetylcholine and nicotine, respectively. This mutation increases the ion flow when acetylcholine or nicotine binds, thereby destabilizing the ion channel's gating region and making the neuron more excitable. This hyperexcitability resulted in mice having a much more amplified neural response to nicotine. These mice included adult males only and their wild-type counterparts. To assess the effects on hippocampus-dependent learning and memory, the wild-type and knockout adult and adolescent male and female mice underwent a pre-exposure contextual fear conditioning paradigm. On day 1, the mice were placed in a chamber and administered either nicotine or saline. On day 2, they received an immediate shock, and on day 3, they were re-exposed to the chamber, and their freezing behavior was measured. Freezing behavior is a natural fear response in mice. Researchers use it as a way to measure both memory formation and extinction. After the initial shock experience, the mice were placed back into the same chamber, but this time with no shock. Over repeated visits, they gradually freeze less and less. That decrease in freezing is what's known as extinction learning, and it means that the mice are updating their memory and realizing the environment is no longer dangerous. Extinction was measured on days 4, 5, 10, and again at week 4, with no further drug administration. The second part of the experiment focused on the hyperexcitable adult male mice. They were first trained to lever press for food, then surgically implanted with intravenous catheters and allowed to self-administer nicotine for 9 days at varying doses until a stable intake was reached. Using increasingly complex ANOVA models, the researchers analyzed how the variable of genotype, sex, age, nicotine exposure, and time interacted with each other to influence fear conditioning and nicotine self-administration. If you're interested in diving deeper into the specific methods or statistical models used, definitely check out the full paper.
So now that we've discussed the background and methods for the experiment, we can understand the significance of the results. The 4 primary implications based on the experiments performed are: Number 1, sex-specific vulnerability in learning. 2, dysregulation of fear, memory, or extinction. 3, the bidirectional control of the nAChRs gene in nicotine reinforcement. And lastly, the interaction of the nAChRs gene during adolescence. By the end of this discussion, we will outline the preclinical indications that alterations in the nAChRs gene can, in fact, make an individual more or less susceptible to nicotine addiction and more or less capable of processing memories of fear, depending on whether the individual is male or female and whether they are an adolescent or adult. So the first result we will discuss is the role of the nAChRs in sex-specific vulnerability in learning. Through the hippocampus-dependent contextual fear conditioning paradigm, it was determined that adolescent female mice do not differ significantly from wild-type controls in their ability to learn fear context, indicating that the knockout of the nAChRs gene does not impact learning for adolescent females. Similarly, knockout and wild-type adult females learned equally well. Adult males, however, did not align with this trend. Adult and adolescent male wild-type mice showed significant greater learning and memory than the knockout males, indicating a male-specific learning deficit in the absence of the nAChRs gene. Based on these results and previous research, this supports the claim that the nAChRs gene is essential for hippocampus-dependent learning in males, but not in females. The biological mechanisms underlying memory formation may rely on different nicotinic receptor subtypes across sexes. The second aspect of the study was the fear extinction paradigm, during which both sexes showed a decrease in freezing between day 4 and week 4. which indicates successful extinction or unlearning of the fear response. For genotype, adult knockout females that were treated with nicotine failed to extinguish and showed increased freezing by the end of the 4-week trial. Adult males exhibited low, stable freezing levels relative to their female counterparts, making extinction less pronounced. Interestingly, nAChRs knockout in adolescent females actually facilitated fear extinction. These results indicate an adult female risk factor as seen in the deletion of nAChRs combined with nicotine exposure, leading to increased fear behavior during extinction training rather than during extinction. nAChRs deletion induces sex, drug, and age-dependent effects on extinction, with female mice susceptible to deficits during adolescence and adulthood, while male mice are susceptible to deficits only during adolescence, but not adulthood. Freezing in adult female mice may be due to spontaneous recovery or the reappearance of a previously extinguished conditioned response after a period of time has passed without reinforcement. It was also determined that nicotine treatment modulates extinction in female adults. With acute nicotine administration impairing fear extinction, it is possible that this behavior is due to increased ventral hippocampal excitability and increased CFOS expression in non-GABAergic ventral hippocampal cells. Adult females also had the highest degree of corticosteroid released when exposed to nicotine, and nicotine may disrupt extinction learning through increased stress response and increased excitability in the ventral hippocampus. Alpha-2 expressing nicotinic acetylcholine are found in the CA1 region of the hippocampus, which could contribute to genotype-dependent effects between nicotine-treated adult females. This has significant implications for understanding the mechanisms behind post-traumatic stress disorder, or PTSD, which is more prevalent in women than men, and is characterized by failure to extinguish fear. Individuals with PTSD may avoid situations or individuals that remind them of a traumatic event, and may show hyperarousal due to a dysregulated fear response, which is believed to be related to dysfunctional learning and memory. In preclinical models, extinction training is used to study the mechanisms of PTSD because there are parallels in exposure-based therapies that are used clinically to decrease pathological fear responses. Since PTSD is higher in women than men, there is a potential role for sex-specific neurobiological mechanisms underlying the conditions. Alpha-2 receptors may be a key factor in sex-specific vulnerabilities to PTSD and anxiety disorders, and findings of this field hold translational and clinical relevance through the demonstration that deletion of the nAChRs leads to nicotine, age, and sex-dependent differences in extinction behavior, which may reflect altered vulnerability to PTSD.
So now we will investigate the significance of the reinforcement in hypersensitive nAChRs mouse lines. As opposed to the knockouts, which were created to explore what happens in the absence of nAChRs, the hyperexcitable adult male mice were modified so that one of the ion channels in the nAChRs transmembrane domain was destabilized by changing just one amino acid, leucine, to serine. This had the effect of making the nAChR easier to open, allowing ions to flow through the channel more readily. And it actually increased ion channel flux significantly when acetylcholine is bound. So this point mutation essentially makes the nAChR exponentially more reactive to nicotine, resulting in the hyperexcitable mouse model. It was first established that both wild-type and hypersensitive mice increased food intake at identical rates, so genetic manipulation of the nAChRs gene did not affect natural reward learning. It was then established that both genotypes reached a stable level of nicotine self-administration between the 4th and 9th days of the experiment, so other than the nAChRs hyperexcitability, there were no observable differences between the mutant and wild-type mice. The hypersensitive genotype earned fewer nicotine infusions during the first 2 days of acquisition compared to wild type, and also showed significant decreases in nicotine intake, specifically at 0.1 milligrams per kilogram dose compared to wild type. Additionally, a previous study used this paradigm to investigate the effects of nAChRs deletion on nicotine reinforcement using adult male nAChRs knockout mice, and it was found that nicotine self-administration was significantly enhanced during the first 2 days compared to wild type. Taken together, these results indicate that nAChRs alterations specifically modulate the brain's response to nicotine, not general reward-seeking behavior. Additionally, genetic manipulation of the nAChRs gene bidirectionally modifies nicotine self-administration behavior, as hypersensitive but not wild-type mice showed decreased dose response at the intermediate dose of 0.1 milligrams per kilogram of nicotine. Knockout nAChRs males adults showed increased initial response, while hypersensitive models resulted in decreased initial response, demonstrating the sensitivity of the alpha-2 containing nAChRs in mediating the rate of initial nicotine self-administration. Individuals with alterations to the nAChRs gene may have increased or decreased susceptibility to the reinforcing properties of nicotine. As animals move from adolescence to adulthood, the role of nAChRs actually shifted. In knockout adolescent females, the absence of nAChRs appeared to facilitate fear extinction, which is possibly due to nAChRs influence on the onset of puberty or circulating hormone levels such as estradiol. This suggests that the alpha-2 receptor may interact with hormonal changes during adolescence. According to previous studies, alterations in estrogen and progesterone levels that occur in females during the onset of menses have been known to affect extinction learning significantly. There was no difference in extinction behavior when females had not yet undergone menarche. Females that underwent gonadectomy before the onset of puberty presented with negligible levels of progesterone and estrogen and exhibited improved extinction recall. Ovarian follicles were also shown to express nAChRs with the alpha subunit detected throughout the ovary. The quantity and type of receptors expressed changed during follicle maturation, with nAChRs exhibiting higher expression in the antral follicle than neonatal ovaries. Knockout of nAChRs in adolescent females may have resulted in decreasing circulating levels of estrogen and progesterone by impacting the antral follicle or delaying the onset of puberty, altering hormonal state during the extinction phase. There may be an interaction between pubertal genotype-specific modulation of learning. For both male and female mice, the immediate shock stimulus occurred before the onset of pubertal hormones as compared to adulthood, and extinction learning was examined post-puberty, which may have underlying consequences for sex-dependent effects observed in nAChRs during adolescence. Ultimately, the nAChRs gene, which encodes the alpha-2 nAChR subunit, does play a critical role and sex-dependent role in hippocampus-dependent learning, fear extinction, and nicotine reinforcement across a lifespan. The authors suggest that future studies should investigate the impact of both nAChRs knockout and nAChRs hypersensitive mouse lines on nicotine self-administration, reward, extinction, and reinstatement behaviors to establish additional parameters that may be modified in nicotine-seeking behavior during adolescence and adulthood.
We've gone over a lot today, so let's take a step back. What does all of this tell us? This study shows that alpha-2 subunit of nicotinic acetylcholine receptor is not just a small structural component of a receptor, but rather plays a larger role in learning, memory, and nicotine reinforcement. Its effects are both age-dependent and sex-dependent, as we see learning deficits in male mice and varying extinction patterns in females. When the receptor undergoes a point mutation that makes it hyperexcitable, we see changes in nicotine reinforcement as the brain responds to nicotine differently. A key takeaway from this study is that these effects depend on age and sex. The interaction between individual genetic makeup and the particular sensitive period of adolescence shapes long-term vulnerability to nicotine addiction. While this study answers important questions, it also raises some new ones. Future studies should examine how alpha-2 containing nicotinic receptors are expressed across development in both males and females exposed to nicotine. Since the medial habenula interpeduncular nucleus circuit is strongly involved in nicotine withdrawal, it would be valuable to see whether selective expression of alpha-2 receptors in specific brain regions contributes in any way to the behavior differences observed. Additionally, future studies should assess circulating ovarian and androgen hormone levels to better understand how sex hormones influence alpha-2 receptor function. Lastly, assessing nicotine dose response curves across varying age and sex groups, along with examining hyperexcitable mutant mice in adolescents and adult females, would help paint a better, more complete picture of how this receptor shapes addiction-related behaviors. Ultimately, this work moves us one step closer to understanding why some individuals may be more susceptible to nicotine dependence compared to others. By identifying specific receptor subunits such as alpha-2 that play such major roles in nicotine addiction, this study helps build a more precise and biologically grounded model of addiction risk. Understanding how sex and age-dependent addiction really is allows therapeutic strategies and recovery to be a lot more informed and effective. If you're interested in learning more about the effects of nicotine across the lifespan, follow us on Instagram at Translational Addiction Podcast for updates, and check out the infographics and papers on the Loftypore Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Loftypore Lab at UC Irvine, exploring how to understand addiction and